All right. Well, good morning or good afternoon to everyone. Thank you for those that are here for attending the Successful Entrepreneurship webinar series that we've been hosting. Today's guest is Corey New. He is professor at University of Colorado in Boulder and also has a startup company in the area of cartilage repair that he's going to talk about today. So, Corey?
Yeah, thanks a lot, Dominic. I appreciate the invite. I'm going to just share my screen, and I want to just confirm that you can see the first slide, Dominic.
Yes, we can see it.
Okay, great. Yeah, listen, thanks for the opportunity to talk about our journey from academia to industry. One thing I wanted to just sort of mention and discuss today is what it's like to be, at least my version, my case, of someone in academia who's trying to commercialize new discoveries in the lab, and some of the challenges that we've encountered. So we have formed about 5 years ago a company called Tissue Form, and you see its tagline there: nature inspired restorative biomaterials. We use materials that are derived, non-synthetic materials effectively, to make what we call like a flowable allograft. And what's important here is that, as Dominic mentioned, I'm a professor at the University of Colorado Boulder. And so we have to navigate this space transitioning from the academic world to industry, and do it in a way that that helps advance the company mission without sort of sacrificing any of our research goals at the same time. So a couple of things that I wanted to discuss today are first the development of what we call a flowable allograft for integrative cartilage repair. And then I also want to, at the same time, really discuss this idea, of a case study, basically presenting our company Tissue Form as a way to learn from a number of mistakes that we've made along the way. The first thing I want to mention is that there are at least three strategies that I've encountered when dealing with trying to bring some kind of a product to market. A lot of engineers I work with and see have encountered over time take this approach that I would sort of characterize as a solution in search of a problem. A lot of people I see say, "Hey, I've got this really cool technology. Where can I use it?" And sometimes there's a good fit with some use case, but oftentimes there's not. And so I'm a little more skeptical of this approach. We try to make products, make new advances in the lab using a problem-driven approach, meaning that we're identifying some kind of medical problem or technical challenge that motivates us to find a solution. And the one of the surprising things in this kind of commercialization journey has been maybe a third category of a market-driven approach, meaning that even if we're addressing an important problem, we may not have the right market approach to really make something that is helpful and useful. So I want to compare and contrast these as I move forward through the next few slides. And maybe first I'll just say where I'm located. I know people are tuning in from all over the world. This is Boulder, Colorado. We're right at the front range of the Rocky Mountains in the United States, in about the middle of the country. In the foreground, in the lower part, you'll see the University of Colorado. Some of these beige sort of pinkish sort of buildings. It's like a sandstone. And in the background, you see the mountain range. It's a really interesting place to live. There is a lot of excitement around startup innovations, not just in med tech or biotech, but in a number of different fields. And I also want to mention that the work that we do in our laboratory portion has been really outstanding with really fantastic people over the last several years. This is not the most up to date. We have some new members that are on the top. And I want to just highlight for a minute Jeannie Barthold. If you see my cursor, if you can't, it's upper left corner, sort of the black and white picture. She's a former student who has been involved with this commercialization journey with me all the time. A lot of the work and sort of decision making you'll see along the way has been as a result of her. Okay, so let's just dive in. And I want to just emphasize this idea of problem-driven approach, something that's motivated us. So I don't think anyone on this call or maybe watching would, necessarily not understand or not appreciate the idea that musculoskeletal health has numerous problems, including arthritis, which is a global problem. Typically, one of the hallmarks is the wearing and the loss of articular cartilage in the knee joint. And I think we can all appreciate, at least in the United States, it's the number one cause of disability. 1 in 4 adults suffer from arthritis, and it affects over 40 million individuals, at least in this country. Of course, we can always think about end stage strategies like total knee replacement. And if we address total knee replacement, they typically, you might say, optimistically would have, for an active person, maybe a 15 or 20-year lifespan, which is outstanding. But if you're a young person, this presents a number of problems. I don't necessarily want a total knee replacement at early stage in my life. So the problem comes in that for the repair of cartilage, especially like, say, focal defects, there are limited treatment options, especially treatment options that we might wish to integrate like a material into the existing cartilage network, this integrative approach. And we've encountered along the way that surgeons typically need flexibility to solve a range of problems. Maybe a surgeon encounters multiple defects in a joint or some other kind of comorbidities. We've taken an approach of using acellular matrix, which I'll describe in a couple minutes, which has initiated this whole commercialization pathway because we think it has potential for broad treatment. Now, what is a flowable allograft? Okay, we're basically leveraging the idea that we would transplant allograft material from one human to another human, except that we put it into a flowable form because sizes vary, and we're trying to free some constraints that are imposed by current allograft solutions that I'll discuss a little bit later. So we're typically thinking about treating focal defects in cartilage, one or more focal defects in cartilage, but with an injectable sort of material that could potentially solidify and provide mechanical and other sorts of functions in a knee joint. Okay, so let me just back up for a minute and talk about early laboratory studies that initiated this whole commercialization approach, right? So starting from the earliest time points, and hopefully you can start to see our decision-making process as we thought about moving toward first in human or product development and so on. So initially we were working with materials that were really, to us, very interesting. What you're looking at is a scanning electron micrograph that was presented and created and collected by someone named Tyler Novak. So he's a former student. He had an initial National Science Foundation graduate research fellowship, a very small seed grant that allowed him some flexibility. And he started to explore research topics. And he started playing around with, taking materials like articular cartilage and morselizing them, mincing them, particulating them and breaking them up and seeing what he could form. What you see here is a false colored, image. The gray sort of amorphous shapes, those are particles of cartilage, and they're devitalized. So you'll see like remnants of cells in some of the locations. And then in the middle, this false color is like a collagen gel that's sort of holding it all together. Okay, so the idea is that we were breaking apart materials like cartilage and then reforming them and recapitulating them in some fashion. And although you see collagen here, we're not really using collagen anymore. We switched to a hyaluronic acid-based approach. Well, what we found early on, just to show you like 2 data slides, okay, early on is that we found, that when you take materials and pack them together, we approach what's called and exceed what's called a percolation threshold, meaning that we don't really need to worry about the properties of the gel. The properties of the minced tissue on the inside dominate the mechanical response. They interact with each other, they bump against each other. So you can imagine that if you had a material like some minced cartilage and then you start to pack them at high density, they start to achieve mechanical properties that are really close to native tissue. And at the same time, one of the other motivating aspects of our early work was to show that when you put cells in the space between these particles, they have a limited but small ability, appreciable ability to crawl back into those particles. They can crawl in, say less than about 700 microns or so. They will crawl back into to these particles. And so if they're distributed throughout this minced matrix sort of mess, they will recellularize an entire construct. And we showed that this process is driven by chemotaxis. So some of the remnant growth factors that are left behind in these particles. Okay, this is all fine and good, and I'm not here to give a science talk, okay? I want to just basically lay the groundwork to say, look, we had these initial discoveries, and it led us to think about pursuing commercialization. This was a turning point, okay? Now, we're sort of backing up and sort of thinking about our approach. Are we now, do we have a solution that we're sort of searching for a problem, right? Are we falling into that trap right from the start? And I asked that, deviating from this problem-based approach, because we were considering this combination of particles, okay, and a way to combine them as a platform technology. So we were envisioning that we could take any tissue, and we have taken many, many tissues, bone, ligament, meniscus, dermis, liver, kidney, the list is quite long. We've taken tissues and we've particulated them. And then we have, processed them in some manner, like lyophilized or devitalized, or left them cellular, and then thought about ways to pack them back together. So we're thinking about then particles that are combined with functionalized macromolecules. This has in our view, broad applicability. And even more so, we've eliminated the need for any kind of synthetic photo-initiated crosslink or any of those things. This is a temperature-mediated process. We just take particles of some form and put them with a functionalized macromolecule, in this case, a functionalized hyaluronic acid. And they form a stable, really interesting construct, okay? So again, we've now deviated slightly from this problem-based approach where we're focused solely on cartilage and thought, okay, getting excited, right? It can be applied in a lot of different areas. And then we realize we need to be a little bit cautious, because any tissue that we select, if it's cartilage or liver, we enter into a number of complications that we didn't expect at the beginning. For example, how do we make this tissue in like a controlled environment so that we could actually transplant to somebody? How do we do it like in a CGMP facility? If we go after every tissue, well, then we're setting up a lot of facilities and a lot of paperwork. So we had to pull back from this concept of being sort of like solution looking for a problem, and really ask ourselves, where is the market, and where are we going with this? So about 5 years ago or so, we formed this company called Tissue Form. This was a company formed with my, between myself and this former PhD student, Jeannie Barthold, whose PhD work you essentially just saw a lot of that recellularization work, and percolation work. And at that time, about 5 years ago, we made this decision to try to go down a commercialization path and explore it. And partly because of Jeannie's interest to help lead the company, we were able to do so, right? I still maintain a faculty position, more so as like a scientific advisor in this venture. So one advantage of living in Boulder, Colorado, is that we have a really outstanding tech transfer office called Venture Partners. And annually, we have, I guess we're really fortunate because we have the ability to apply for all sorts of funding specifically for the creation of new companies, things like the Lab Venture Challenge, the New Venture Challenge, which are many are supported in part by the Colorado, state funds, basically, and what's something called the OEdit. And there, CU Boulder the tech transfer office is extremely active. Many, lots of patent activity each year. Many startups coming from all parts of our campus. And one thing we were asking ourselves at that time was, was this the right time to start Tissue Form? I guess, in some respects, looking back, maybe the answer is no. Because we were early stage. We're still considered early stage. We're a preclinical company, seed-based company right now. We have not yet, we don't yet have approval for for the materials we're talking about. We don't have a product on the market yet, but we are far along in the sense that we've completed a lot of animal studies. At that time, though, we really didn't even have much of a market fit analysis that had been conducted. So I think it's fair to say that we were maybe it was not necessarily the right time. However, we were presented with funding options, that we could sort of move forward. So we took the plunge. I guess there's a good chance that we could have failed at some point by now, and luckily we haven't so far for I think a number of reasons. Some of the challenges we encountered at that time, and even really still kind of persist, are should I keep my faculty position? Should I completely divest from the University of Colorado and move to this sort of startup role? And I say that because I know of university systems in the United States where that really is kind of a primary option or a main need, that you have to have this kind of separation. And luckily here at Colorado, the startup involvement is really appreciated and encouraged. And so I didn't need to do that. I could maintain my position as a faculty member, which gave our team the ability to apply for funding sources, non-dilutive, grant-based funding sources. Um, and it also, with my position, this is common, at least in many places in the United States, where we have a certain number of days a week or in a month where we're able to work on activities completely outside the university. So I was able to leverage that activity. And then, of course, there's the effort from Jeannie Barthold, right, my co-founder. Other issues we encountered were, how do we form a team? How do we take the two of us, two scientists, and build out a lot of needed knowledge around that, around just the idea of forming biomaterials. A lot more is required to advance to, some kind of commercialization effort. And what are the natural next steps? So in the area of team formation, initially we had just the two of us, myself and Jeannie. And about, maybe a year or so later, we added Juliet High. She's just about to finish a PhD in our laboratory. So she really took on this effort of manufacturing materials in a CGMP sort of environment, which we do now with a partner, which I'll just mention in a minute. And we're looking to hire another scientist to help us because we're completing a number of benchtop studies that are required for our commercialization effort and regulatory effort, I should say, through FDA approval. We have really fantastic contractors and consultants, groups like Nelson Labs and Colorado State University. Colorado State University, if you're not aware, is one of the best large animal facilities in the United States. It's a fantastic vet school. It's just really close by. It's within an hour's drive of where I'm sitting now. And, they routinely do large animals, goats, sheep, horses, common large pivotal animal models for orthopedic repair and commercialization. So we're fortunate they're here. If we weren't living in Colorado and our company was located somewhere else, Florida, United States, Canada, whatever, we probably would want to come to this institution anyway. So we're fortunate to be nearby. We work with regulatory partners. Our knowledge, has grown over the years. We're not experts, and so we had to surround ourselves with a group from Proxima. Excuse me, and then we also partner with Allosource. Again, we're fortunate, very fortunate that we live in Colorado, and that Allosource, one of the major providers of allograft tissues in the United States and worldwide, is located maybe within another hour, from where I'm sitting. So Allosource, will receive donor tissues within, just a few hours of death and create viable tissue products. Maybe one leg will yield 200 different medical products. Despite that, despite creating products like, prochondrics and dermal grafts and things like this, they still, unfortunately, throw away most of the donor tissue. So the concept that we can come in and extend the gift of donation and take this, what would be discard tissue, but otherwise healthy tissue that maybe didn't qualify for allografts, and particulate it and create like a flowable material is very appealing to Allosource. So Mike Florin is someone we work with there. And then as we've, had continuous sort of support financially, we've had, a number of people at a local finance office, Richter, here in Boulder. Additionally, we've surrounded ourselves with some really important say KOLs. Okay, so Rachel Frank is one of the leaders in joint preservation surgeries and approaches, and she's been a long partner since she arrived here in Colorado in a number of studies, and it's been a natural process for us to work with her. Tyler Novak is at Gilead in the Bay Area of San Francisco. He's a former student. He's the one who did the initial particulated material studies in the laboratory so many years ago, and he's maintained involvement with us. And Nicole Glaros provides a different perspective, right? She's formerly with Techstars, and she's really an expert in raising funds, and so helping us as we transition toward moving from non-dilutive funds to actual investment funds. So, I guess the point is we've had to surround ourselves with a lot of individuals, okay, to fill in gaps where we do not have any kind of expertise, and we rely on these people heavily. So finding these partners has been essential for us, and we're grateful to everyone we work with. One of the questions I asked just a minute ago on a former slide was, and what are the next steps? So not knowing necessarily much about market fit yet, and as scientists, and at the early stage of forming this team, we really just dove back into the science. Okay, we sort of just thought, well, we need to translate this science into a product. Okay, what should we do? How do we move this forward? Well, we were reading papers that, we had done these benchtop studies. Next logical steps were to move into animal studies. This is something that is maybe very common, or, something you would expect from many people who are listening. But for us at the time, we were very unsure of how we should move forward. So we took this leap of faith that there would be some kind of a product-driven, approach, so a problem-driven approach that we would want to solve. And about that time, we were fortunate to receive a grant from the CDMRP, so the Congressionally Directed Medical Research Program, to conduct a large animal study. So to take the next step. And it was really well timed. It was also, sort of an easy sell. We have this really strong, need worldwide for cartilage repair. People who are, working in the military or related to the Department of Defense have, abnormally large, prevalence of problems. And so we were thinking that we would move this product forward in a way to help those individuals. So with this funding, we were able to do quite a lot and advance in really profound ways. First of all, we really honed in and defined composition, a certain combination of tissue, matrix, and gel that could be delivered. Okay, so what I mean is it could be flowed into a defect with an open procedure or arthroscopically. And after it is inside the joint, then it would solidify and bond and connect to the surrounding cartilage tissue. So we envisioned that we would have this sort of two syringe approach that at the point of delivery, on demand, the surgeon would mix the combination, the tissue and the gel together. It would initiate a cross-linking reaction. And so within that time window, you could inject this into a joint and promote repair. And the interesting thing about this to us is that the process is what we term integrative. In other words, the material bonds to itself and it bonds to the surrounding cartilage in about, say, 15 minutes or so. This is a nice working window with inside that works well in the workflow of a surgery. So now I'm going to show you, this is an image from Colorado State University and one of the many goat patients that we operated on. For those of you who are familiar with the university, Jeremiah Easley is a great surgeon. He's on the left-hand side. He's doing this procedure for us in combination with Brad Nelson. And what we do is effectively, at least for this procedure, is we created sort of like a augmented microdrilling or microfracture procedure. So we effectively remove the surface cartilage in what would be a model of a defect, a cartilage critical size defect. We drilled into the subchondral space to access the marrow. The surgeon mixed the material right at that time of surgery and injected it into the joint space, into that hole. And we did it as an open procedure, at least at this initial stage. And then he just simply smoothed and contoured the material to fill the defect. It was very simple. He then started closing up the joint immediately, and the animals were up and completely weight bearing within about 45 minutes. So this procedure was all very sort of fast and easy, and it was one additional step effectively in mixing and delivery right at the point of doing what would otherwise be like a microdrilling procedure. So this material that we injected, this was our first large animal study. Okay, this was a porcine-derived cartilage. Okay, so we took cartilage from pig, not human yet, took it from pig, and went through this procedure. Now we have gone through another iteration. We have a human-derived version of this, which we're hoping to start a pivotal animal study within the next 12 months. But the issue is that, or what we see that as this product is having advantage is that when we've compared like what we call microfracture or microdrilling and controlled contralateral limbs, we often see that there is a fibrous response as well documented. You know, a lot of type 1 collagen is inside of the repair tissue. In contrast to this material, our particulate on the right-hand side, something we've been terming naturalized, we often see that we always see that it's filled with type 2 collagen. There's a healthy amount of proteoglycan, aggrecan, and the surface contours look really excellent. So when we're moving forward, we're envisioning this as a like-for-like replacement, the flowable human tissue that's been repurposed from donors that's effectively creating a like-for-like solution. This is a very flexible kind of procedure. In other words, if we see multiple defects in a joint, you can simply fill them. It's all natural matrix. I haven't talked a lot about the material, but I have just mentioned that it's effectively tissues that have been oftentimes devitalized or decellularized, and then combined with a functionalized hyaluronic acid, no additional cross-linkers, no photoinitiators, nothing like that, which makes the procedure really simple, low cost, and it's a one-stage procedure, unlike, for example, a MACI. Okay, so we've advanced now to this point of doing, a first large animal study, which has had really successful results, and we're sort of writing that up. My science side tells me I still want to write this up and publish it, but we've been thinking more about really identifying the market fit. Although we might be solving a problem in the laboratory, we haven't really yet said, does anyone really care to buy this thing? Why should we go forward? And we can do animal studies and iterate on new formulations and new benchtop studies until the end of my career, probably. But how do we actually move it into a first in human study or, clinical trials and so on? So we've transitioned the strategy from problem driven to more market driven. And I want to say that a lot of our shift in a market driven approach has been motivated by our involvement with the tech transfer office here at Colorado, which has sent us on a path down to be heavily engaged with the National Science Foundation I-Corps. Now, if you're outside the US and you're not aware of this I-Corps, I think there are some mimics of this around the world and some other variations in different countries, but effectively, this is a federally sponsored program that has a mission to accelerate translation of scientific and technical discoveries. What they do is provide entrepreneurial training. They literally give us funds so we can go do this customer discovery at conferences. We went to the sports medicine conference last year. We went to a veterinary medicine conference, multiple conferences, so we could interact directly with potential customers, potential users, and try to understand what is the market? Is there a market? Are we just sort of spinning our wheels? And it has been an eye-opening process for me. I went in so reluctant because it's a lot of work. You have to do over 100 interviews. You're spending all time traveling around and talking with people. But I realize now that is probably the most valuable thing we've done in the 5 years we've been together as a team in this tissue form venture. If you don't have access to the I-Corps, you can find a version of this effectively with this book called The Mom Test. And I recommend you find it and you get it. What I like about this is that just read the quote from the back, right? Everyone is lying to you. They say you shouldn't ask your mom whether your business idea is good because she loves you and she'll lie to you. In other words, your mom loves you and will lie to you. So the idea here is that you have to learn how to ask questions to to figure out what the market is. If you go in and say, "Look, I've got this great technology. How are you going to use it? And how is it going to transform your, clinical practice?" Then you sort of miss the point. You have to go in. What I've learned is that you have to go in and ask questions like, you know, what problems are you encountering in the clinic? What's your number one problem right now? What challenges do you see? What would you need to transform your clinical practice? And if you're lucky, you might realize that the solution that you have is the right fit, and then it's a perfect alignment. Or you might realize that it's not, right? And we have to pivot, change the technology, go a different direction. So when we went through this process of learning through the I-Core and through by going and talking to a number, tons and tons of people and startup, established companies and contract research groups and all sorts of things, we realized actually it was eye-opening again. We discovered a really a nice critical gap. We realize that surgeons are having a difficult time treating individuals who are 30 to 35 to 50 years old. There's a debatable standard of care. It's sort of all over the place. Different people have preferences and I think there are some good solutions like MACI is one good solution. Allografts, viable allografts is another good solution, but they're not without their problems, right? So MACI is extremely expensive and it's a two surgical surgery approach. Um, Surgeons are reluctant to use it for a variety of reasons. Allografts, you know, allografts are in limited supply. They're a fantastic approach, but we learned even that some patients have to wait up to a year once they're diagnosed to get one of these things. Like there are definitely limitations with these existing procedures, and we found through all of this effectively where we fit in. We're not expensive. We're on par. We're in a direction of like allografts, like from something like Allosource. We are not scarring. In other words, like products like BioCartilage that some of you may be aware of, and even de novo are typically recombined with a fibrin glue. So we're using like non-native cartilage materials to try to hold some tissues in place, which often can lead to scarring. And so we feel like we're in this space. We think we're in this space where we're promoting healing, and we're also really cost effective. Like we're trying to keep the costs really low in general, so that we are able to make this a part of a surgeon's workflow. Take an existing procedure and add on maybe one more step. Not transform the approach too much, but provide outcomes that are potentially transformative for the patient. And this led to what I would say is maybe one of the last sort of aspects of this is that some unexpected challenges arose. Okay. So unexpected challenges through our customer discovery process were downstream of any of the science that we would have initially predicted. And they were in the area of regulation and reimbursement. And so we realized, like looking at, for example, MACI as a case study, that we could have a potentially really excellent procedure, and yet the cost can be prohibitive. How is it that we're going to convince a hospital system where we maybe do or don't have a reimbursement code? MACI does, but if we don't have a reimbursement code, how would anyone even buy this thing? Do we need to generate a code? Do we need to sort of leverage other reimbursement codes in our U.S. healthcare system so that the surgeon and the hospital system can be paid? Are they going to be operating at a loss every time they do a procedure? So then that would limit the scale. A lot of issues arose around this idea of reimbursement that we did not expect. And we also didn't really appreciate when we were starting out the regulatory process. So the regulatory process in the US, most places is stringent and for good reasons. And the idea of taking a product to market is not like, especially in orthopedic repair and cartilage is not an overnight process. It's a multi-year step, and we have to have the patience to see it through to the end. And it really helped us understand the formulation of our material. How do we say minimally manipulate cartilage and particulate it in such a way that we combine it with a minimally functionalized material so that there are, although we're doing a sort of a combination type product, that these materials are not really outside of the standard of what's already being provided. This is an ongoing challenge, and it's something that informs our strategy going forward, right? So we have this timeline. We are currently, right now, we have, established with some active funding from the NIH and NSF, we've established a CGMP manufacturing pipeline, and we're conducting all of these check boxes for biocompatibility and toxicity studies through some contract groups. So we now effectively have a really well-defined process where tissues that are not being used for viable allografts are given to us, and we take them through a series of steps in our clean room facility, and to the point where we were able to have a product that we can distribute and put on a shelf or send around. And we're going through this kind of necessary testing currently. And it also informs like the directions going forward, because if we look at where we might anticipate being on market for cartilage repair, it is like, say 6 years, good case scenario for us from now. And that involves targeted funding for non-dilutive funds, right? Through like, pivotal animal studies, SBIR, continuation kind of SBIR funds to conduct, say, a study in horses, and doing then ultimately transition toward first in human trials before filing for PMA and so on. So part of this strategy then informs a shift from maybe a comfort zone for someone like me in the scientific research space to doing certainly looking for doing pitches and looking for venture funding that will help accelerate us to the end. At the same time, because this is a long pathway, we've recognized that platform technology has some earlier exits and some offshoots, right? We could, having completed the biocompatibility studies, within about a year from now, we could at the make our approach sort of a parallel and go after something like a wound healing response or dermal fillers or bioink development. These are all possibilities, and we have to like be careful, okay? Because we have to ask ourselves, is our mission cartilage repair, and do we see a market? Yes. It's long, so the temptation is to want to spin off and say, okay, let's focus our effort in other areas. For example, going after like a dermal filler or bioinks. And so we become like that solution looking for a problem, which is tempting, but also a potential major hazard. However, I would say I would rely back on our recent approach with like the mom test and the iCore, and it's allowed us to really rapidly evaluate markets, like, in a really quick sense. So within the last about 2 weeks, we've taken that iCore approach and we've interviewed a number of additional people, about 30 more people from pharma and from contract groups. And we've recognized that the bioprinting direction is not a viable direction. Bioprinting is generally driven now by the sale of printers. But most people who are using bioinks or buying bioinks, they're really not. They're maybe like researchers like me in a laboratory who are making my own bioinks. So why should I necessarily buy another one? The market is really small. And now things may change over time, but we feel like we have the tools now to evaluate things quickly and change and adjust and pivot as needed. So right now, we're in the process of identifying a number of surgeons so that we can, after we progress through this pivotal animal trial, move toward a clinical safety study, first in human study in people, in patients, and set us up for PMA approval and along a very long and interesting FDA regulatory pathway. So maybe I'll just say in the end here, did we choose the right path? What are the lessons learned? I guess I would say, we may have started a bit early, but at the same time, we had opportunities to do so. So I guess the answer is yes. Did we choose it? I guess we did the best we could, given our limited knowledge. Okay. And I think that some things I've appreciated is that we've found strength as a lean startup, very number of actual sort of paid employees. We've been able to really fluidly move between a lot of different educational opportunities. incubators like NSF, ICOR, creative destruction labs out of Toronto, Octane out of Southern California, local groups here like Innisphere and the Rocky Ventures. We've been able to raise a lot of non-dilutive funds that have helped us approach all these benchtop and animal studies. We're not planning to get stuck in this sort of like reformulate, redo animal studies for the next, 20, 30, 40 years. We are actively trying to say, well, how do we leverage this formulation that we feel is at the CGMP sort of manufactured level and move it toward human trials? We've had initial conversations with potential strategics. We're thinking, what is the end game here? How do we ultimately leverage distribution channels to make this more of a mass market approach? And we've been able to establish really strong partnerships, especially with Allosource, where we've set up shop and effectively are doing a lot of our clean room work. It's been a great partnership, and we look forward to that continuing. And then, finally, I just want to say there's a lot of strength in learning from mistakes. Okay, I think that we initially started as a problem-driven approach, and we moved ultimately to a market-driven approach. Maybe, and now when we evaluate new applications of what we're doing, we think we more closely look at the market-driven approach. That seems to be a better indicator, transitioning from academia to, at least in our experience, from academia to industry. And we definitely avoid taking a solution and trying to search for a problem. It's so tempting. It's so tempting to take that approach. But we have to actively sort of say, okay, no, look, is it really addressing a problem? What's the market say? We've learned along the way to define our customer, in different applications. Is it the surgeon? Maybe not, actually. Maybe it's the hospital purchasing board. Maybe it's another company that we want to exit to. So learning who the customer is helps define that market-driven approach. And we've really taken advantage of customer discovery. What questions help me understand what the product market fit is? And we found that really has helped us accelerate our understanding of how to advance commercialization. So we're not yet in humans. We're not yet on the market. But this approach we've had so far has helped us to get to this preclinical stage, and we've got a lot of momentum, and we're really excited about that. So I just want to say thanks for the opportunity to talk. And this is where I'll stop. And yeah, thanks for your time, Dominic.
Corey, that was really insightful and an excellent presentation. Thank you so much. What an exciting journey, right?
Yeah,
So
thanks a lot. Appreciate
I,
it.